So welcome to the day 2 of Learning with Ease. And today's topic is RNA interference because it is one of the most chaotic topic among the students. So suppose you wrote very important on a piece of paper and you don't want it to be seen by anyone else. So you just covered it with another piece of paper so that no one sees it. And that's how the RNA interference works. A very familiar example of this has been given in the NCERT textbooks, and that is of a very dangerous villain, which is a nematode, a Meloidigyne incognitia. You must have heard this name, which basically attacks the roots of the tobacco plant and causes the decrement in its productivity. And to deal with this problem, we use the RNA interference. So what we basically do is to find the most important gene in the nematode's body so that it forms a life-saving protein, say protein X, in his or her body. If that protein X doesn't forms in the nematode's body, then the surely nematode will die. So we will take two genes with such types but with opposite polarities. And why with opposite polarities? Because with opposite polarities we will get the two mRNAs which will be complementary with each other. Now, after the transformation process into the same tobacco plant, we will be getting the complementary mRNAs. अब हमारे पास जब complementary mRNA है into the same tobacco plant, तो जाहिर सी बात है they will stick together. But when they will stick together, तो हम दोनों को पता है कि complementary mRNA जब आपस में मिलेंगे तो वो double stranded बनेंगे. और double stranded RNA कभी exist नहीं करते, ठीक वैसे ही जैसे तुम्हारी वो exist नहीं करती है. ओके तो जब डबल स्टैंडर्ड mRNAs या फिर RNAs बनेंगे, सो जाहिर सी बात है द डाइजर विल कट देम इनटू द टाइनी मॉलिक्यूल्स ऑफ RNA. Along with that, the risk, that is RNA induced silencing complex, will be stick to those RNA molecules, tiny RNA molecules. Once we are done with all of these, now if that particular nematode attacks that particular tobacco plant, then those tobacco plants which are having those tiny RNA molecules along with risk molecules will get into the body of that particular nematode. So once that specific gene undergoes the process of transcription, it will form an mRNA, right? So once that mRNA is formed, those tiny RNA molecules which are actually the complementary with that formed mRNA will stick to that particular mRNA and make it double stranded. और एक बार वो डबल स्टैंडर्ड अगर बन गया, दैट मीन्स इट्स प्रीटी ऑब्वियस टू से दैट इट विल बी श्योरली डिस्ट्रॉयड बाय द डाइजर. और वंस इट इज डिस्ट्रॉयड बाय द डाइजर इनटू द स्मॉल पार्टिकल्स ऑफ आरएनए एक्चुअली, दैट मीन्स अब वो प्रोटीन, लाइफ सेविंग प्रोटीन एक्चुअली वो नहीं बनने वाला नेमाटोड के अंदर. और अगर वो नहीं बनेगा देन नेमाटोड इज श्योरली गोइंग टू डाई. और वंस इट इज गोइंग टू डाई, दैट मीन्स अब हमारे टोबैको रूट्स के अंदर रूट नॉट्स नहीं बनेंगे और इसकी प्रोडक्टिविटी जो है वो इंक्रीज करेगी.